This is Mada from Translational Imaging Group of Center for Medical Image Computing and Center for Advanced Biomedical Imaging at UCL. I'm going to present our recent work about sublayer thickness estimation of mouse cerebellum cortex. The cortical thickness has been widely used as a quantitative morphological analysis for neurodegenerative diseases such as Alzheimer's disease or Down syndrome, both in clinical and preclinical settings. However, studies has been limited to the cerebrum and little attention has been paid on the cerebellum. The image on the right demonstrated the morphology of a mouse cerebellum. It consists of foliar lobes separated by fissure lines, similar to the sulcus and gyrus in human cortex. There are three anatomical sublayers in its gray matter: the molecular layer, the Purkinje layer, and the granular layer. So the aim of our study is to derive a framework to estimate the thickness of the molecular layer and the granular layer, and this is achieved through extraction of the middle Purkinje layer. This slide shows the pre-processing steps. Firstly, the cerebellum is extracted using multi-atlas label fusion. The gray matter is then segmented by optimizing the Gaussian mixture model of tissue intensity. The fissure line is extracted using divergence-based skeletonization. We adopted a Laplacian-based cortical thickness estimation to get the distance mapped towards the inner cortical surface as well as towards the outer surface. We also parcellate the gray matter into function-aware subregions for regional thickness statistical analysis. The Purkinje layer is extracted in two steps. Firstly, we applied a modified French's filter to enhance the contrast of a planar structure. Areas with significant filter response are used as initial estimation. To extract the Purkinje layer to its entirety, we need to further extrapolate the remaining missing part from the initial estimation. We project the ratio between the inner cortical surface distance and outer surface distance onto the initial estimation. We then applied 10 Gaussian smoothing with exponentially decreasing kernel size to propagate the average ratio onto the entire gray matter. Voxels with minimum distance between the original and the smoothed ratio were included to create the final extracted Purkinje layer. The thicknesses are defined on the Purkinje layer and averaged for each segmented function-aware subregions. The distance to the inner cortical surface are defined as the granular layer thickness, and the distance to the outer surface are defined as the molecular layer thickness. The proposed method has been assessed on a dataset of gadolinium enhanced ex vivo T2 MRI scans of mouse brain, which includes 14 wild-type mice and 14 transchromosomic mice, which models Down syndrome. Both the voxel-wise thickness map and the regional average thickness shows that the transchromosomic group have thinner gray matter thickness as well as sublayer thicknesses. The difference becomes significant after normalizing to the total intracranial volume. As a validation, these results agree with findings from previous studies. So, in conclusion, here we presented an automatic framework to estimate the cortical sublayer thickness of mouse cerebellum, and the sublayer thickness measurements were enabled by a two-step extraction of the Purkinje layer. The validation on ex vivo MRI data suggested a reduction in regional average thickness in mice model of Down syndrome. Thank you for your attention.